everyone. Welcome to our webinar today. I'm really happy to introduce Dr. Ingrid Simonik from Addenbrooke's Hospital, part of the Cambridge University Hospitals in the UK. She actually presented this topic at the ASHG annual meeting just a month or two ago, and we're really happy that she's able to present this here for those of you who could not attend the conference. So she will be talking about data filtering and prioritization in NX clinical software with a retrospective analysis of intellectual disability referrals that she has done. I'm going to hand it over to her so that we can begin. Dr. Simonik?
Good day, everyone, and thank you for the introduction, Shalini. I work as a consultant clinical scientist at Cambridge University Hospital, and I specialize in diagnosis of rare disorders. And in my department, we use clinical exome sequencing using the TrueSight1 expanded panel from Illumina to complement all our other methods and tests which we use for the diagnosis of rare disorder, particularly the array CGH, the MLPAs, and the Sanger sequencing. So we all know that whilst the steps from sequencing to variant calling have been standardized for all types of sequencing referrals in our lab, we are aware that we use different methods for variant annotation, filtering, and prioritization depending on the reason for referral and the panel size. And about 18 months ago, we validated the use of Nexus clinical software for an integrated analysis of CNV, SNV, and absence of heterozygosity from our NGS clinical exome data for intellectual disability referral, which currently is our largest panel used in the lab. And just to quickly walk you through to what we're currently doing when we are looking at this data in NX Clinical. So first of all, we examine all the copy number events in NX Clinical using the probes and the B allele frequency tracks in the software, and we do that for all the chromosomes. And once we completed that and annotated all our CNVs within Nexus Clinical, we then follow it by filtering all our SNV variants, and we do that currently by requested panel, which in our case of intellectual disability referral is going to be intellectual disability panel, that's how we call it, and or DDD G2P panel, and that again will depend on the reason for referral. And then we also use filtering by other means, for example, by the event types, interest level, and variant consequences. And we then follow our sort of data filtering by CNV and SNV classification and annotation, and we record all that in NX clinical table view. So about a couple of months ago, in June, I decided to complete a service evaluation of our latest intellectual disability referrals into the lab. And I did that because I felt that we may improve the effectiveness and the efficiency of analysis if we perhaps use other methods for data filtering and prioritizations. So in order to do that, as part of this service evaluation, I completed sort of a comparison between our data filtering using the predominantly sort of intellectual disability panel filtering, and I compared it to a filtering of our data using the HPO terms within the NX clinical environment. And I also evaluated the use of subscores within NX clinical for prioritization of the data which we received after the HPO filtering. And the subscore is essentially a p-value like probability, which is assigned to a gene, and it's based on the strength of its association with the phenotype, and this is all included within sort of the NX clinical platform. So whilst I was performing this survey's evaluation, I became aware of certain limitations within sort of the way we're analyzing the data, but also the sort of with regards to the data source. So, I mean, it became very clear that our intellectual disability referrals are a very heterogeneous group of referrals with a significant proportion of these cases having a very non-specific phenotype. And this will have an effect on filtering using the HPO terms. Furthermore, I mean, we are currently using, for our integrated analysis, we're currently using a single pool of control samples for CNV calling, and this is obviously not ideal. Ideally, what you would want to use is sort of almost like a matched control sample from within the same run. So the same rules apply for array analysis as they apply for NGS analysis with that regard. And then finally, I mean, when I examined the data, I realized that I was not using the latest HPO annotation within the NX clinical platform, and this again had an effect on the results of this study. So first of all, I will walk you through to the way we currently analyzing the data. And those of you who are familiar with the NX clinical platform, you would sort of be able to sort of recognize some of these images from the sort of the NX clinical view. So once you load the data in, you can open the data, and the first thing you will see is this screen. It's like a whole genome view with all the annotated variants being displayed as these little lollipops next to the sort of the ideograms of the individual chromosomes. And our sort of standard analysis pipeline will essentially, we will go through the sort of the probes view for each of the chromosomes. In this case, the probes view is for chromosome 20, and you can see it's sort of the distribution of the probes is nice and tight, so the data is very clean. And what we see here is an apparent gain around the centromere on chromosome 20, and using this CN event track, you can click on these little triangles and you will see that this particular gain and all the other ones are present in many other samples, and you can disregard this as essentially a benign variant or an artifact of an array. And we analyze each of the chromosomes like this, and we record all the rare variants and then annotate them within NX clinical. And we also look at the BRL frequencies, and with the normal distributions, I mean, in this particular case, there is a small regions where there is an absence of heterozygosity on 20q, and we may want to record it, but at the moment we don't really record it. Unless we know that the families are consanguineous and we need to look, for example, for homozygous variants, etc. So that's the first part. The second part is we, following the CNV analysis, we then perform the SNV analysis, and we start with the basic filtering of the data based on the quality and population frequency. So we tick all the relevant boxes we need to think. We don't really tick off all the sort of high frequency variants in individual populations, because our referral population is varies with regards to their sort of the population background. So we don't want to lose any significant variants by removing them. We then follow by filtering by event consequences. We check the interest level filters, and these are set. So these filters are preset, and we always use the same sort of setting for all our sample analysis. And we then also check for similar previous cases within our software. So for example, this in-frame deletion, as you can see here, it's not, it doesn't record any population allele frequency in the software, but it's clearly present in our data set. So this can be excluded from further analysis as a benign variant or a spurious call. And following the basic filtering, we are able to reduce the number of calls, which in clinical exomes we will normally get about 40,000 annotated variants, and by applying the basic filters, we can reduce the number of these variants by 20-fold to about 2,000. And to further reduce the number of variants, we use our standard diagnostic panel filtering, and we have some panels included in our NS Clinical, which includes our ID panel and the G2P panel. And depending on the referral and what the patients consented to, we will apply one or both of those panels to our data. And just a quick information about the panels which we use. So the intellectual disability panel is based on the green gene list, which is curated by Genomics England, and this is the link to the web page. And obviously, because we're using the TrueSight1 expanded panel, we don't sequence all the genes which are on the green gene list, and the overlap between these green gene lists and our clinical exome panel is about 400. Well, it's 444 genes, not about 444 genes. And then we also use the G2P panel, which is curated list of genes compiled by the clinicians as part of the Deciphering of Developmental Disorder study. And again, you can find the link here, and you can download the panel from the website as well. And the overlap with our TrueSight1 panel is 1,127 genes. And as you can imagine, there is a definite overlap between these two panels, which is 634 genes. And what is nice within NX Clinical is that if you, for example, filter your data by intellectual disability panel, for example, and then you are asked by the clinician to reanalyze the data using this panel, all your annotation for the ID panel for the overlapping genes are still going to be there when you're displaying your G2P. So it makes the reanalysis much faster this way. And I mean, depending on the consent and also the family situation for our referrals, and the filtering may be performed by genotype. For example, for consanguineous families, we filter the data based on homozygous genotype in the proband, and sometimes we are asked to filter the data by loss of function variants, so by the consequence as well. So using this sort of standard analytical pipeline, the data which are included in this sort of service evaluation, I mean, consists of 108 clinically reported variants in 98 probands. So in those, so I included 211 singleton referrals, and in those we reported 108 variants. And these variants consisted of 91 reported SNVs and 17 reported CNVs, and they were reported in two types of referrals, which I explain a little bit later. And it was 25 intellectual disability referrals and 73 intellectual disability plus referrals. And I mean, 66% of those reported variants were classed as VUSs, and we use our national guidelines for classification and reporting of the CNV variants and ACMG guidelines for as well as our national guidelines for reporting for classification of the SNV variants. And as you can see here, 25% of the reported variants were classed as pathogenic, i.e. class 5 variants. And I also looked, as part of the service evaluation, I looked at the inheritance of some of these variants. I mean, because we are analyzing singletons, we, as part of our standard reporting practices, we may recommend parental follow-up for some of the variants. And I mean, by looking at the sort of the location of the variant, as well as the genomic location of the variant, as well as the sort of inheritance, I can see that 20% of our reported variants map to chromosome X, and 18% of the variants are de novo, and 5% of the reported variants are homozygous variants in consanguineous families. And we're left with about half of the variants not being with unknown inheritance, and and some of these are being followed up sort of subsequently. And I mean, one of the reasons why we would recommend a VUS, for example, to be followed up, is to be able to reclassify the VUS either as likely benign if inherited from a normal parent, or as likely pathogenic if identified de novo. And we followed up a VUSs and reclassified them as likely benign, and 3 further VUSs were reclassified as likely pathogenic as part of this sort of follow-up investigations. So moving forward to sort of looking at the HPO sort of virtual panel filtering of the data as an alternative to filtering of the data using the intellectual disability panel or the G2P panel. So I mean, this screen just quickly shows you how you can actually enter the HPO terms within the NX Clinical platform. So you can click on this button here, and this particular sort of like is a sample attributes screen will appear, and then you can enter your phenotype in this bit by pressing the edit button. So once you press the edit button, you get this phenotype screen where you can type your phenotype, for example, developmental delay, and then you will get this menu of sort of developmental delay HPO terms, which you can click. In this particular case, the patient was referred for delayed speech and language development, and we take this, and developmental delay, which was classed as severe or profound, and you can again click here, but I usually also click on this sort of more general term up there when I enter the data. And once you enter the data, you apply this, save the changes, and that way you can sort of you can annotate the phenotype. And I did that for all 211 samples which were included in this service evaluation. And what I spotted quickly was the limitation, which I mentioned earlier, is that for a large proportion of these referrals, the only thing which we received on a referral card was either global developmental delay or intellectual disability or some sort of autistic behavior. So I classed this group of referrals as intellectual disability, and this is the typical sort of phenotypes box I sort of had for many of these referrals. And then there was another group of referrals which included other sort of clinical features, and the most common ones were seizures, microcephaly, I mean dysmorphic features which are entered as abnormal facial shape, and then there were delayed milestones, hypotonia, overgrowth, obesity, etc. And this is the sort of like typical sort of phenotype screen which I got for this group of referrals, which was assigned as ID+. And as you can imagine, when you so just to give you an example, so when we apply the intellectual disability panel to our sort of 2,000 variants after the basic filtering, we will get about 40 variants per sample. When when using this sort of filtering. So when I use the HPO virtual panel filtering for example, just sort of ID referrals, I got 90 variant calls. So clearly, I mean, you don't really want to manually examine 90 calls. What you like to do is to somehow prioritize these calls so the analysis can be much faster. And I use this subscore to prioritize these calls. But going back to the HPO versus intellectual disability panel filtering, I mean, what I discovered in those with regards to all the reported variants was that there were 2 variants which we reported in the 211 samples, but they were not identified either by the ID panel or the HPO virtual panel. And one of them was a class 5 variant, and it was in this particular gene, and this gene was reported as intellectual disability gene in 2019. And the reason it was actually identified originally and reported to the patient was that we were asked to use the transcript overview to identify the loss of function variants and whether or not in essentially unfiltered sort of variant screen after the basic. So no, in the screen before the ID panel was applied, and this is how this variant was identified originally. So then we were hoping that by HPO virtual panel filtering, we will identify all the reported variants, but actually that wasn't the case. So 9 reported variants in 8 genes were detected by the ID panel, but they were not detected by the HPO panel filtering. And I'll explain this. I explained this a little bit later. And one of those genes in 2 patients, there were 2 different variants in 2 patients in this gene, which is called NAA15, and that was published as a OMIM disease gene in 2018, and then it was C-NOT3 gene. It's in another example. It was a frame shift variant in this particular gene, and this gene has only been assigned as an OMIM disease gene last month. And then what I found that when I use the HPO virtual panel filtering, it was better at detecting actually CNVs compared to the ID panel genes. And I mean, the reason for it is very simple: is that with the HPO, I mean the variants are being the genes are being assigned to the phenotype based on the associations within the HPO database. And in this case, the patient was referred for dysmorphic features, and it seems the HPO picked up the partial deletion of this gene, TCF12. As you can see, it's very clear, this partial deletion. And TCF12 is autosomal dominant craniosynostosis 3 gene. But because this gene is not on the ID panel, obviously this call by ID panel filtering was not identified. So it's missing in here. So there were 6 such cases in our data set. And there were 5 reported SNVs which were detected by the HPO panel filtering, but not by the ID panel filtering. And again, the reasons are quite obvious for that. So, for example, this patient here was referred for obesity and all these other features, including moderate global developmental delay, and a loss of function variant was identified in this gene called SH2B1. And as a cytogeneticist, I recognize this gene immediately because it's one of the genes which is deleted in this recurrent 16p112 deletion syndrome. And this deletion syndrome has been associated with body mass index quantitative trait locus. And we know that this gene is one of the more commonly sort of mutated genes in people with obesity, even though it is not assigned as a autoimmune disease gene, but within the HPO is associated with obesity. So that was one, and the second one, just to show you, is a homozygous single base pair deletion in this gene, ZFY-V26, and this patient was referred for profound global developmental delay, as you can see here, and there were no other clinical features. And this particular condition is autosomal recessive, and it is associated with variable developmental delay. So this is a homozygous pathogenic variant, and it was identified in an individual with sort of consanguineous parents. So, and finally, I think I would like to show this case because it sort of illustrates very nicely the usefulness of the sort of the integrated CNV, SNV, and absence of heterozygosity analysis. And this case was spotted immediately. As we opened it, we saw this whole genome view, and there were loads of purple in here. And this purple indicates that there is an abnormal B allele frequency profile. And as you can see here, the profile was abnormal for all the autosomes. And what I mean by abnormal is that so this B allele frequency distribution has got like 3 clusters normally along here and then in the middle of here. But in this case, there were like 5 clusters, as you can see here. 5 different clusters, and this is actually consistent with chimerism in the blood sample from this particular patient. And this patient was 4 years old. And what was interesting is that, as you can see here, there is no purple on the X chromosome, and there are these 2 blocks of yellow, which indicate absence of heterozygosity. And you can clearly see it here. So there is absence of heterozygosity for the B allele frequency, but the proband is a male, so he only has one X chromosome. So how come that we have a heterozygous pattern for the B allele frequency here? And the answer is that actually in 50% of his cells, he has 1 X chromosome inherited from the mother, and in another 50% of the cells is the other maternal X chromosome, which is present in this proband. So, I mean, obviously, as you can imagine, I mean, when you have a chimeric profile in your sort of sequence data, it is very difficult to analyze and look for heterozygous or homozygous rare variant, which would be pathogenic. But interestingly, we did identify using our ID panel filtering. We identified this probably damaging VUS in MECP2 gene, and that was detected by HPO and all the other filtering means. But it was disregarded by the analyst because if this variant was pathogenic, I mean, you can imagine in male, the male would not be viable because that's normally the case. But because this patient is a male but has got 2 different X's in his cell, he essentially mimics a female profile. So this variant could still be pathogenic. So it was reported as a VUS, and it was followed up. We received a sample from saliva for this patient, and there was no chimerism in saliva, and this variant was present in hemizygous state. And I mean, this was this patient had a fraternal twin brother, and he shared his one of his genotypes from in the DNA from the blood was shared with his twin brother, and the twin brother did not have this variant in the MECP2 gene. So we still don't know whether this variant is likely pathogenic or not, but we are following this up further this family, because there are several other affected individuals in this particular family as well. So this was an interesting case, and as I mentioned before, I did do HPO filtering in all other patients, not only the ones which included in this service evaluation, not only the ones which had a reported variant, and I did identify other variants which are pathogenic or likely pathogenic, and these were in 3 different genes and in two different individuals, and this is now being further followed. There is a further follow-up has been initiated because these variants have not been reported previously in these patients. It is. I will discuss this within the conclusions, but clearly there is no right way to filter the data. The panel filtering using the ID panel or the G2P panel has its advantages, but so does the HPO filtering. But what I was interested in, because we are essentially assigning, or we are essentially filtering more variants using the HPO for our very non-specific and heterogeneous group of referrals, I was interested to see whether using the subscoring metric, which is included within the NX clinical package, we will be able to more effectively prioritize all these variants. So this metric is it's called the significant associated with phenotype or sub score metrics. And I'll just use this definition. It's a p-value-like indication of probability that any gene is associated with the patient's phenotype by chance, and that means that the lower or more significant the p-value is, the genotype phenotype, the more significant the genotype phenotype association will be. So in our data, the subscores were assigned to a large proportion of the reported variants, and for sort of the further analysis, I essentially classed these variants into 4 categories based on their significance, and these are the 4 categories. And you can see majority of the reported variants will have the subscores which are in this sort of range of significance. So they're not very highly significant. It's only 3% of the variants reported were in this high significance range for the subscores. So what does it mean? So we without knowing precisely what the subscore values were, we managed to observe essentially that the ID referrals, which are the more heterogeneous referrals, the subscore values were less significant. So these are the ID referrals indicated with sort of the little red diamonds, and you can see here they are clustered in this sort of less significant range of the sort of subscore values. And then the second thing which we observed is that, intuitively we would expect the class 5, which means pathogenic variants, to have higher subscore values, but that's definitely not the case. So the distribution of the sort of the subscore values is not affected by the class and the pathogenicity class of a variant, because it is essentially just the association with the gene, not the variant, which we are assigning the significance value to. So there were a few key bets with regards to the subscoring in my service evaluation. First of all, I did not include our CNVs in these, even though you can subscore the CNVs, and it works really well because we used a pooled sort of control samples instead of matched control samples to each of our samples. There were quite a few samples where with lots of false positive calls. So the prioritization of CNVs using the subscoring wouldn't have worked. So we only applied the subscoring to our SNV variants. And you remember when I said that there were some SNV reported SNV variants which were not detected by the HPO virtual panel filtering. Well, all these variants will not have the subscores assigned to these. So it's only 79 out of 91 variants in that study which had the subscores associated values, and 12 variants in 9 genes failed to annotate, and they were assigned a default value of 1. And these were all the genes, and again, including this one, which I mentioned before, and this one, which I mentioned before. So we now know that all of these genes would have a subscore using the latest HPO annotations in NX Clinical. So this is a very important point. So because we didn't use the latest version of the NX Clinical and the latest annotations, we failed to score these variants. But what is more interesting and what is highly valuable, when we use the subscoring on the remaining 79 SNVs, what we found that all the reported SNV variants ranked in the top 10 variants in our table view after prioritization and after the removal of low confidence scores. So you will have some remaining low confidence score in your table and some calls which are present where the minor allele frequency is quite common in some of the sort of the population, individual population. So, so as you can see here, 35% of the reported SNVs ranked in the first position. So this is pretty good, and scoring seems to be working really well. So what have I learned from this service evaluation? Well, I learned that a lot of the referrals which were included in the service evaluation had a load of reanalysis, and I mean load of it's 2 and more reanalysis performed. So there were 2 and more reports issued for these probands because different types of panels have been requested on the same referrals. So on the retrospective evaluation of these referrals, I can now see that the combined variant filtering using the ID and the HPO virtual panel testing would increase, would not only detect all the reported variants, but would also increase the number of reportable variants by 10%. So that is quite a significant increase. And then, as you can imagine, once you increase the number of variants which you filtered for a certain referral, it becomes more tedious to manually examine all those variants, and this could be overcome by using the prioritization tools such as the subscoring within the NX Clinical software. So this may improve the effectiveness of variant prioritization, providing that the sort of the local installations of NX Clinical are continuously updated. So that's my learning point. And in the end, I would like to thank some of my colleagues who are involved in the sort of the intellectual disability analysis in the lab. And I would like to thank Shalini for all her help and support with subscoring and the use of various tools within NX Clinical. And I will be happy to answer any questions you might have. Thank you.
Now we will go over to the audience questions. In avoiding this variant fatigue, what is the time saving with this process?
Avoiding the variant fatigue. Yes. So I think currently, I mean, when we apply these various data filtering tools within NX Clinical, we can analyze a case within sort of 20 minutes.
Great, thank you. How often do you update the HPO terms in the software, and how often do the genes on the ID panel get updated?
Right. So we don't know how often the ID panel gets updated. I mean, we usually check on a monthly basis whether or not there are updates available on the Genomics England panel app webpage, and then we would update our ID panel if there is any difference between what we're currently using and what they're proposing to be used. Because we haven't been using the HPO filtering as part of our diagnostic workup up to now, we haven't been really careful about updating, but it is my understanding that the HPO is updated several times a year. I don't know exactly how many times.
Yeah, currently in NX Clinical, the annotations are updated quarterly, starting next year, and well, less than a month. Basically, it'll be updated on a monthly basis.
Yeah. I mean, it's clearly very valuable to use the latest updates because we seem to be hitting the latest genes.
Yeah.
Quite often. Yeah.
You showed an example of a gene that was not detected by either workflow, but in the table you showed it was identified as pathogenic. Can you explain this?
Yes. So in this particular case, we were asked to filter by loss of function. So when you're filtering your data, you can filter by various parameters, and we were asked by the clinician to filter by loss of function variance, and we detected this loss of function variance, which was rare, and it was in a gene, and there was no OMIM entry assigned to the gene, but because we checked manually the OMIM, we could identify that this was a new OMIM disease gene, and yeah, so we managed to report it that way. Yeah.
Okay. Great. Multiple gene panels were used. How did you apply them? And did you need to reprocess the data?
Right. So we did not need to. So within NX Clinical, you don't need to reprocess the data when you apply different panels. You just have to have your panels entered into NX Clinical. And that is a very easy process. It's very easy to do. And currently in our lab, we have various panels. So for example, when a patient has got microcephaly, learning disabilities, and other clinical features, what normally happens is the clinician will ask for microcephaly panel first, which is a small panel of genes associated with microcephaly. And if we don't find a pathogenic variant using that panel, then the clinician may ask for the intellectual disability panel, and if no pathogenic variant is identified, then they can ask for the G2P panel, etc. So many of these patients which are looked at, they have, as I explained, they have very heterogeneous sort of complex phenotypes. So many of them will have several reanalysis performed on the data. But within NX Clinical, you wouldn't have to essentially input the data again and again. You just have to filter the existing annotated variants within NX Clinical using different panels.
All right, thank you. And then the last question here, should previous cases be reviewed on some periodic basis since the data in OMIM, et cetera, is constantly updated? Should it be only unsolved cases or all cases?
It is a very good question. Yes, we currently don't have sort of guidelines with regards to reanalysis, whether we need to reanalyze everything using the updated sort of versions of the resources which we have available. But because up to 5% of the of our complex referrals will have more than one diagnosis, and very often on the first time around, you won't identify the genetics causes for all their, which would explain all their clinical features. My feeling is that it would be easier to actually reanalyze all our samples on a periodic basis, just to see whether or not, I mean, unless you have identified a clearly pathogenic variant, which would explain all the clinical features in the patients, I would suggest to reanalyze it. But currently, we are only reanalyzing when requested by the clinical teams.
Okay. Great, thank you so much. That's it for our questions. Actually, there's one more that just came in. Why don't we address it? We have time. What is the overall percentage of no findings?
An overall percentage of null findings. I'm not sure I understand this question correctly, but essentially, I mean, we reported, I mean, out of the 211 individuals which are included in this evaluation, I think in 98 individuals we reported a variant, or a couple of variants. So I think a large proportion of our referrals, we haven't identified pathogenic, likely pathogenic, or a variant of unknown significance which were reportable. So it's between 50 and 60% of our referrals we don't have variants reported.
Great, thank you so much. That's it for our webinar today. Thank you everyone for attending, and thank you so much, Dr. Semenik, for having these two sessions.